Okay, thank you, and good afternoon. So today I'm going to talk about my research plan on this topic, the environmental fate and ecological implications of plastic litter, specifically microplastic in macrophyte system. My name is Hoi Shing Lo, and I work as a postdoctoral researcher at ACES. So this is a little outline of this presentation. So first, I will introduce you the macrophytes as pollution hotspots of microplastic, and how plastic litter or microplastic affect macrophytes to this point, to this moment, is unclear and unknown. So we aim to investigate the fate of microplastic in macroalgal bed in terms of retention of microplastic and as an entrance to the food web as well. So we will also try to assess the microplastic effects on the plant by its metabolite production for antifouling, as well as the potential effects of leachate from microplastic. So I will give you a very general introduction of microplastic. So what is microplastic? It is a tiny particles, typically defined as smaller than 1 millimeter or 1,000 micron, made of synthetic polymers. So, and there are two types of microplastic. The first one is primary microplastic, and second one is secondary microplastic. So, primary microplastic is something designed and manufactured to be that small, smaller than 1,000 microns. And secondary microplastic is the result of fragmentation of larger pieces of plastic that is released in the environment. And maybe, for example, under radiation of UV, as well as the mechanical abrasion in the ocean, so that it breaks down the plastic into tiny pieces. So we observed a lot of plastic in many organisms in almost all trophic levels, from invertebrates, fish, and even mammals. But the effects and toxicity to this point is much debated and quite unclear. This, for example, depends on mainly the diverse nature of polymers. We have different kinds, different types of polymers, polyethylene, polypropylene, for example, and they tend to aggregate with lateral particles. So if they aggregate, the surface properties, the physical chemical properties will differ. And some organisms have been evolved to deal with particles, not only microplastic, but also lateral particles. So, for example, filter feeders, mussels, they have an ability to get rid of the unwanted particles by producing pseudofeces. These examples create uncertainty to understand the underlying effect mechanisms. So in the Baltic, the pollution level is considered to be low to moderate when compared with other parts of the world, like where I came from, Hong Kong. The Asian shorelines are much more polluted in general. So it is estimated that the pollution level, you can see that, not quite serious, but microplastic is still there. So why do we concern macrophyte? So first, Baltic has these types of macrophyte system, seagrass meadows that you can find in Sostera marina in the west coast of Sweden, and macrophyte bells, Fucus, vesiculosus, coastal meadows, and reed beds. They provide an important ecosystem services and functions. For example, they are very excellent primary producers. They transform in organic carbon to organic carbons, and they are all biodiversity hotspots. So they provide nursery habitat, feeding grounds for a wide range of organisms, from microscopic algae, fish, invertebrates, and they all produce organic matter in the form of detritus. So the organic matter could be buried in sediment for quite a long time. But they also trap material from water. So not only sediment, this could be also applied for microplastic as well. So microplastic in macrophyte beds of Baltic, to this moment, to our understanding, there are two studies concerning these particular topics. So the first one is here, was done in Estonia. So they collect sediment inside the seagrass beds and outside the seagrass beds. So they found much higher microplastic in the sediment inside the beds when compared with the vegetated sites. The similar results has been obtained for the macroalgae beds as well, but this time they collect water rather than sediment. So similar result, microplastic in water is much higher than outside the water. So a very similar result has been obtained other regions of the world, for example, the macroalgae in the US, seagrass in China, and seagrass soils along the coast of southwest Spain. So it is clear that macrophyte can retain microplastic, but the key question is, first, what is the main driver of microplastic retention? And second, what are the effects of microplastic on macrophyte? So it has been proposed that there are two potential mechanisms for retention of microplastic. And the first one is the enhanced sedimentation rate. And my previous study, this published data, collected live species of algae, 100 individuals from different sites, and there are low significant difference between sites. But when we classify macroalgae into different morphologies, in this case, filamentous and non-filamentous algae, we find much higher microplastic inside the algae, inside filamentous algae, than the non-filamentous algae. So the surface topography, the space between the algae itself, could enhance the sedimentation rate so that you can find more particles, not only microplastic, inside filamentous algae. And the second potential mechanism is the adherence of sticky surface. So yes, has been demonstrated the periwinkles, the gastropod here, when they move, they do locomotion, they left behind a pedal mucus. So if the microplastic particles in contact with the mucus, they tend to stick there and stay there, accumulate there. So we ask the question, how about biofilm on macrophyte surface? And this inspired our plan, the first experiment. The biofilm composition or abundance affects the microplastic retention. So in the following experiments, we plan to use the Fucus vesiculosus bladder rag as a model organisms to represent macrophyte because first, they are abundant, they are well studied and used widely in biomonitoring. So the main composition or main constituent of biofilm is something called EPS, the extracellular polymeric substances. And they are the constituents of EPS in turns is, for example, the carbohydrates, protein, DNA, and trace amount of humic substances. And we propose that this production of EPS is controlled by the bacterial community and abundance. So we try to control that so we can also control the EPS content or composition of that as well. So we will find out the correlation between EPS content, composition, abundance, concentration, and microplastic retention capacity of macroalgae. So the follow-up study is that if the biofilm really can retain microplastic, will it be the entrance point to the food web? So we plan to collect 3 different types of animals. They are herbivores, detritivore, and biofilm grazer. So classify them according to their feeding habits to find out which types of an animal is the most affected. So if so, the energy transfer efficiency as an indicator to support a food web will be assessed. So we want to see if it still can support a food web by using isotope labeling. So I've talked about the microplastic retention and potential entrance to the food web, but we also want to see from the seaweed, from the macroalgae perspective, will the microplastic affect the physiology of the seaweed as well? So Fucus, in this case, is known to produce metabolite for first, antifouling, and second, chemical defense, herbivorous defense. And to control the metabolite on the seaweed, the Fucus evolved to control the bacterial abundance and composition as well. Well, one thing to understand this is the microplastic contain a totally different microbial composition and abundance than the surrounding environment. So will the presence of microplastic and the associated microorganisms affect the production of metabolite as well? So this is the question we will ask and we try to figure out. And this is a summary. So we learned that the macrophyte is pollution hotspot of microplastic. So we aim to investigate the fate in terms of retention and entering the food web in macroalgal bed and try to assess the potential effect. And more experiments will also be designed, for example, the effects of leachates as well. So if you have any question, feel free to ask, or maybe you can email us. Thank you.
Thank you.